In today's lecture, we are learning about the Renin-Angiotensin System, Juxtaglomerular Apparatus and the acid-base balance. First we see the Juxtaglomerular Apparatus. What is the Juxtaglomerular Apparatus? It is the apparatus and it is the region of the nephron where the afferent arteriole and the distal convoluted tubule come into the direct contact. The main function of this apparatus is secrete the renin by the juxtaglomerular cell. Now see the structure of nephron. This is the glomerulus. This convoluted tubule is called the proximal convoluted tubule. Then U-pin-like structure, it is called the loop of Henle, and the final part of nephron, it is called the distal convoluted tubule. There are arteries in the kidney which called the afferent arteriole and the efferent arteriole. This is the afferent arteriole which carry the blood toward the glomerulus and the efferent arteriole which carry the blood away from the glomerulus. Now see the cross section which show the juxtaglomerular apparatus. This is the distal convoluted tubule and the afferent arteriole. These come into the direct contact and form the juxtaglomerular apparatus. The dark brown shell which is present between the afferent arteriole and the distal convoluted tubule, it is called the juxtaglomerular cell, and the function of this cell is secrete the renin in the blood. Now see the macula densa cell. The macula densa cell, it is located in the kidney in the part of the glomerulus and the distal convoluted tubule also. The cell which is present in the glomerulus, its main function to maintain the blood pressure and the filtrate rate of the glomerulus, and the macula densa cell which is located in the distal convoluted tubule that detect the sodium concentration in the fluid. We see the function of the macula densa cell is regulate the BP and the glomerulus filtration. So, if decrease the blood volume, that means decrease the blood pressure, that is the decrease the blood supply to the kidney. So, the less blood is filtered to the kidney. Due to this, the solute concentration is also decreased. At that time, the macula densa cell of the distal convoluted tubule, it acts as a chemoreceptor to decrease the solute concentration, and it sends the signal toward the juxtaglomerular cell, and the juxtaglomerular cell it secretes the renin in the blood. Now by this process, by this way, the juxtaglomerular apparatus is the work and it secretes the renin. Now we see the renin-angiotensin system. In previous slide, we see how the renin is secreted by the juxtaglomerular cell. The function of the renin-angiotensin system is also the maintain the blood pressure, means the main function is the increase the blood pressure. We see when the blood volume is false or the blood flow to the kidney is false, then the juxtaglomerular cell in the juxtaglomerular apparatus of the kidney which secrete the renin, and the other side the angiotensinogen which is the protein produced and secreted by the liver cell. Then this renin is act enzymatically on the angiotensinogen to release the angiotensin first. The angiotensin converting enzyme is act on the angiotensin first and convert it into the angiotensin second. The main component of the RAS, that is the renin-angiotensin system, is the angiotensin second, which rise the blood pressure. Now the angiotensin second rise the blood pressure in the two way. First, the angiotensin second itself It is a potent vasoconstrictor. So it raises the blood pressure by increasing the systemic vascular resistance. And the second way is it stimulates the secretion of the aldosterone. The aldosterone is the hormone secreted by the adrenal gland, which is located on the apex of the kidney. The function of this aldosterone is increase the reabsorption of the sodium and the water, and that's it increase the blood pressure. This way the renin-angiotensin system is act. So the main function is increase the blood pressure. Now the acid base balance. Your blood need a right balance of the acidic and the base compound to function properly. This is called the acid base balance. Your kidney and the lung work to maintain the acid base balance. Now we are learning only about the kidney and the acid base balance by the kidney. The normal pH range of the blood is between the 7.35 to 7.45. A condition in which the blood pH is below the 7.35, it is called the acidosis or acidemia. And the acidosis is caused the depression, CNS disorientation. In some cases, the patient may be died due to the acidosis. And the condition in which the blood pH is the higher than the 7.45, it is called the alkalosis, and it is also called as the alkylemia. And the alkylemia is caused the over excitability of the CNS and the PNS, means the central nervous system and the peripheral nervous system. In the metabolic reaction of the acid base balance by the kidney, the metabolic waste product produced by the kidney, it is the non-volatile acid and such as the sulfuric acid. The mechanism the only way to eliminate this the huge acid load is to excrete the hydrogen ion in the urine. Given the magnitude of this contribution of the acid base balance, it is not surprising that the renal failure can quickly cause the death due to the saturation of this sulfuric acid in the body. Now the acid base balance mechanism in the kidney. First, the sodium and the hydrogen antiporter, in which the hydrogen is secreted in the urine and reabsorb the sodium in the blood. See, this is the lumen of the renal tubule, and this is the peritubular capillary network, and this is the sodium and the hydrogen antipode. By this antipode, the sodium is reabsorbed in the blood and the hydrogen is secreted in the tubule. Now the three mechanism is based on the secretion of the hydrogen ion in the blood. We see the first, sodium and the hydrogen antiportal. Second is the proton pump that secrete the hydrogen into the tubule fluid. Then third, chlorine and the bicarbonate ion antipode. Now see here, carbon dioxide and the water form the carbonic acid. That carbonic acid is dissociated into the hydrogen ion and the bicarbonate ion. This hydrogen ion secreted in the urine and the bicarbonate ion is reabsorbed in the blood. We see this the mechanism. The hydrogen is secreted in the renal tubule. Then this hydrogen is combined with some component and form the complex, and it excreted in the urine. First, the hydrogen ion is combined with the ammonia and form the ammonium ion. And the second, the hydrogen is combined with the monohydrogen phosphatase ion and form the dihydrogen phosphate. By this way, it is secreted in the urine.